Your body does not regenerate like Wolverine. It was never designed to. Yet millions of doses of a peptide claiming to accelerate healing are already being injected without a single large human trial confirming long-term safety. So, what exactly is happening? In the Marvel universe, Wolverine heals from gunshots in seconds. Bones reform, flesh seals, no scars. In real biology, healing is slow, fragile, metabolically expensive. Muscle tears repair over weeks, ligaments over months. Some tissues never fully recover. Constraint is built into the system. Then came the internet's latest biohacking obsession, the Wolverine peptide. The molecule at the center of this craze is BPC-157. Influencers call it a regeneration accelerator, a tendon repair shortcut, a gut healing breakthrough, a performance enhancer, an anti-aging tool. Some claim it heals torn ligaments in days. Others say it repairs nerves. Some say it reverses organ damage. If a small peptide could safely amplify regeneration across multiple tissues, it would be one of the most significant biomedical discoveries of the century. Pharmaceutical pipelines would reorganize overnight. Orthopedic medicine would change. Post-surgical recovery would collapse into weeks. They haven't. So, why isn't it approved? Why isn't it in hospitals? BPC stands for Body Protection Compound. It was isolated from human gastric juice in the 1990s by researchers in Croatia. The synthetic version, BPC-157, is a short chain of 15 amino acids. 15. For comparison, insulin has 51. Growth hormone has 191. Although it is frequently discussed as an injectable compound, oral use is also popular in biohacking communities, with claims of gastric stability. However, human pharmacokinetic data remain limited. In rodent studies, BPC-157 appeared to accelerate tendon healing, reduce inflammation, promote angiogenesis, new blood vessel formation, protect against certain tissue injuries. Mechanistically, it seems to influence nitric oxide pathways, VEGF, signaling fibroblast migration. In plain terms, it may increase blood supply to damaged tissue and stimulate repair cells. That is plausible biology, nothing supernatural. But plausibility is not proof. Here is the first structural weakness in the narrative. There are no large randomized placebo-controlled human trials demonstrating safe, reliable regenerative effects. None. Most data comes from rat models, cell cultures, small observational reports. Rodents are not humans. Nearly 90% of drugs that show promise in animal studies fail in human trials. History is full of promising molecules that collapsed under controlled validation. If BPC-157 were truly transformative, elite sports medicine and orthopedic practice would already reflect it. They do not. That gap matters. In the United States, BPC-157 is not FDA approved for medical use. It is sold as a research chemical. That classification creates a regulatory gray zone. But regulation is not the only issue. Quality control is another. Products marketed as research chemicals are not manufactured under standardized pharmaceutical oversight. The risk is not only whether BPC-157 works, but whether the vial contains exactly what it claims. Peptide synthesis impurities, residual solvents, or heavy metal contamination may pose risks independent of the intended molecule. In some cases, impurities are biologically more consequential than the peptide itself. This layer is rarely discussed. Let's separate biological plausibility from exaggeration. Reasonable, localized modulation of growth factors may enhance healing in specific tissues. Angiogenesis can improve repair environments. Short peptides can influence signaling cascades. Exaggerated, full ligament regeneration in days. Systemic organ rejuvenation. Universal anti-aging effects. Neurological repair without controlled trials. Healing is tightly regulated. If you amplify growth signaling indiscriminately, you don't only increase repair, you increase biological risk. Uncontrolled angiogenesis is also a hallmark of tumor biology. This does not prove carcinogenicity. It demonstrates that biological systems are bidirectional. Enhancement is never isolated from consequence. Let's quantify the gap. In animal models, some tendon studies reported 30 to 50% faster recovery metrics versus controls. Impressive. But in humans, we lack long-term carcinogenicity data, pharmacokinetic mapping, dose response, safety curves, large cohort outcomes, randomized controlled trials, zero large-scale human validation, and yet injections and oral use continue globally. Without phase-validated safety frameworks, that is not cutting-edge medicine. It resembles large-scale self-directed experimentation. History is not kind to medicine that outruns evidence. There is another variable. Healing is influenced by sleep, nutrition, load management, rehabilitation, intensity, and belief. Pain perception is neurologically modulated. Expectation alone can alter inflammatory signaling and pain thresholds via cortical regulation pathways. If someone heals in 6 weeks instead of 8, was it the peptide, disciplined rehab, natural recovery variance, regression to the mean? Humans assign causality quickly, but correlation is not confirmation. Peptides are relatively inexpensive to synthesize, difficult to patent if described in literature. This creates a peculiar economic structure, limited incentive for billion-dollar pharmaceutical trials, strong incentive for gray market distribution and influencer marketing. In competitive sports contexts, regulatory and ethical considerations also become relevant, even when clinical evidence remains incomplete. The more dramatic the testimonial, the higher the demand. But testimonials are not data. In evidence-based medicine, anecdotes rank near the bottom of reliability, above speculation, below controlled trials. Here is the intellectual fork. Option 1. BPC-157 possesses meaningful regenerative properties in humans that remain underexplored. Option 2. Its human effects are modest, context-specific, or partially placebo-amplified, amplified by online narrative dynamics. Both are logically possible, but only one aligns with current evidence. Right now, the strongest claims exceed the strongest data. That asymmetry matters. The real question is not, does it work? It is. At what magnitude? In which tissue? At what dose? Under what conditions? With what long-term risk profile? Science is scalar. If BPC-157 improves tendon healing by 10%, that is clinically interesting. If it regenerates organs, that is revolutionary. Current evidence supports, at best, the first scenario, and only in animals. Zoom out. If a cheap peptide could safely enhance multi-tissue regeneration, professional sports would look different. Orthopedic recovery would look different. Surgical timelines would collapse. They have not. It is worth noting that a small number of limited or preliminary human observations have been published in specific contexts. However, these remain narrow in scope and have not been validated through large, randomized, placebo-controlled trials. Silence in large-scale medicine is rarely accidental. That silence is data. Wolverine is fiction. Human biology is constraint. BPC-157 may influence healing pathways. It may one day earn a clinical role, but today it exists between promising mechanism and proven medicine. And that gap is where hype grows fastest.